don't get intimidated by the title. It won't be as bad, I hope, as it sounds in terms of getting into details. I wanted to just kind of give you a bit of an overview about the work that we've been doing. And like some other people have mentioned, we got involved in SLC6A1 Connect really because Amber hunted me down. And when she did, it was really great because it was a long time ago when I was a postdoc. It was actually just a couple years after the Human Genome Project started. We were collaborating a bit with the group that had cloned GAT1. Now it's called SLC6A1, Henry Lester and Baruch Kanner. And at that time, no one had really expressed it and done any electrophysiology with it yet, and we were in a position to do that, so we went ahead and used a technique that we've used a lot, and I'm going to talk a little bit about how that works. It's essentially using a system called Xenopus levis oocytes, and these are oocytes we can collect from frogs. They're tiny, tiny eggs of the frog, and we can inject RNA into those oocytes, and then they will express whatever gene that you transcribe the RNA from. And so you can see what happens when you do that. If you record currents across the oocyte membrane, when you apply GABA, you get that downward deflection, that trace, and you don't see that in oocytes that aren't injected with the RNA. And what that downward current reflects, as we heard about earlier, is actually movement of 2 sodium ions and a chloride ion along with GABA. And so that allows us to record a current, and in real time we can measure the transporter's activity. So one of the first things we wanted to do was to try to get some insight into the mechanism by which the S295L mutation causes its disease effects. And so we just did a simple experiment to co-inject RNA encoded or transcribed from a wild-type SLC6A1 gene together with the mutant RNA in oocytes. And what we could see was that the mutant RNA by itself didn't give rise to any transport current at all, and it didn't give rise to any radio-labeled GABA uptake at all. And it also didn't have any effect on the wild-type RNA transcript's ability to induce a GABA current and GABA uptake. And so this kind of gave us some reassurance that it would be a good strategy to try to find ways to upregulate the wild-type copy of the allele of the SLC6A1 because it wouldn't be likely to be inhibited by the mutant allele. And we had been working with a company called Ionis and their foundation and Lorem to do similar studies with other genes. And so it was really exciting to start working with SLC6A1 on this. And it was also a really great advantage that the transgenic mouse line had been developed that was the same knock-in mutation that was expressed in human patients. And what we started to do was to use mouse brain from these transgenic mice and measure radioactive GABA uptake into the mouse, into the mouse brain tissue. And to do this, we would make 300 micron thick slices of brain and just incubate those slices with radioactive GABA and then count the radioactivity after 10 or 12 minutes. And you can see that this just shows the radioactivity that goes into those slices of brain tissue over time, and we can really track where we are in the brain when we do these uptake assays, and we can cut the brain symmetrically in half. These are horizontal sections. So we can do experiments like add drug to one half but not the other to investigate the effect. So the first experiment we did was to, and these are results that are actually very in keeping with other data that you've seen today. This is basically a graph showing how much the total radioactive GABA that goes into a slice, and this is this shows the difference between wild-type brain tissue, heterozygous tissue, and tissue from homozygous 295L mutant mice you can see that as you go from left to right, that the amount of GABA uptake goes down. And the reason I'm showing you this is to basically give you a sense of what it means when you have a wild type activity and then a heterozygous activity, which is intermediate between the wild type and the homozygous activity on the right. Also, what's kind of interesting and a little bit surprising for us was that if you look at the total amount of GABA uptake that's lost in the homozygous mutant, it's really only about 30% of the total amount of GABA. And so this is interesting because, and also I think it's important in considering what's going on in the brain and guiding therapeutic development, because it means that there are other GABA uptake mechanisms in the brain. And I'll talk a little bit about that in a minute. So in other words, loss of SLC6A1 doesn't mean that you lose all the GABA uptake activity in the brain. If we kind of probe into that a little bit more deeply, what's plotted here on the right is actually the amount of GABA uptake that's blocked by drugs that we know selectively block SLC6A1. And you can see that in the wild type brain, there's a bigger amount of GABA uptake that's blocked by this SLC6A1 selective blocker. And again, there's an intermediate effect in the head. And then in the homozygous mutant, there's no uptake at all that's seen, which is actually just what we would have expected, that all the SLC6A1 is missing in the homozygous mutant animal. So it kind of confirms the fact that the mutant's actually behaving like we think, but it's not all of the GABA uptake. Now there are, we've heard a little bit about other GABA transporter gene variants in the brain that are expressed. One of them is GAT3 or SLC6A11. And just like this SKF drug that blocks GAT1 or SLC6A1, there's a blocker that's selective for SLC6A11 called SNAP. And you can see in this experiment, what we've done is taken brain tissue from homozygous mutant animals where there's no SLC6A1 activity, and that you see is confirmed because there's no effect of SKF on the uptake of GABA, because all the GABA SLC6A1 transport is gone. But what's interesting is that when we apply the GAT3 blocker, SNAP, there's a pretty large effect that's telling us that SLC6A11 or GAT3, I'm sorry for using all these acronyms, somebody said something like when they first received a diagnosis that they were really confronted with like an alphabet soup of things like GAT1 and SLC6A11. So I'll just try to stick to SLC6A1 and SLC6A11. So compare what we see here to what we see in the wild type brain. And what you can see here is what I showed you before. SKF blocks about 30% of the total uptake in the wild type brain. SNAP, the GAT3 or SLC6A11 blocker, doesn't block any GABA uptake, which is completely different than what we see in the homozygous animal. But what's odd is that if we add SKF and SNAP together, that there's a super additive effect. And at first this really puzzled us. We thought that probably what was happening was that maybe SLC6A11 was upregulated in these mutant animals in sort of compensating for the loss of SLC6A1. But to make a kind of a long story short, and I'm not going to go into the details too much about this, but there's a complex effect in an interaction between these two different GABA transporters in the brain. And I think it's really important that everyone becomes aware of it. And actually some of this data was out there in the literature, but I think it's been kind of ignored because it's really difficult to understand. This is an experiment done by a colleague at UCL. This is in wild type normal rat brain, looking at the interaction of these two selective blockers of GAT1 and GAT3. And what they see when they do this experiment is that if they add the two blockers together, there's a bigger effect than the block of either one alone. And this, we're right now kind of in the midst of doing some modeling studies and looking more carefully at how this can happen. And hopefully we'll have some answers and some hypotheses to test soon. But the takeaway message is that it's not, SLC6A1 doesn't work by itself in the brain. There are other GABA transporters. Actually, there are at least 3 transporters, 2 in addition to SLC6A1. I'm not going to talk about this data too much, but we think that there's also a lower affinity GABA transporter in the brain. Now, you saw some data Kirill presented about using tools to block GABA A receptors in order to get some information about the level of ambient GABA in the brain. And we've done similar experiments. In our experiments, and these are in, it was great. Dennis showed a picture of the blind man and the elephant. And I think it's funny because I was thinking about showing a slide like that because scientists do use that a lot. We were looking at a certain age of animal in these experiments, and we're recording from neurons in a certain place, and the results may be really different depending on the age of the animal, depending on what the brain region is. But what we find is that in B is showing what we can, the GABA ambient, the GABA A current that's active tonically, that reflects the GABA concentration in the brain of the animals. And the important thing about that figure on the right is it shows that GABA is increased from the wild-type animal on the left to the heterozygous to the homozygous. And we can logically make some deductions about the effect of the SLC6A1 S295L mutation from this experiment. And I'm not going to go through the logic, but what we can conclude is that in the heterozygous mutant animals, GABA levels are approximately doubled in the brain in the extracellular space. And a little less than doubled, sorry, about 70% increased. And in the homozygous mutants, they're a little bit more than double. So that's kind of a mundane answer, but it's, I think, really important. It means that although it seems like a big effect for GABA to be increased by about 50%, it's actually a pretty modest effect. We've also done some experiments to record from neurons and look at these synaptic GABA A currents, and I won't go through the details of this, but our takeaway is similar to what Kirill talked about, with some minor differences that may be due to things like age differences and brain region differences. But the takeaway, one of the important takeaways, I think, is that in the mutant animals, the miniature iPSCs, that reflects synaptic machinery, GABA being packaged in vesicles and being released and activating receptors on the postsynaptic side. That signaling is really pretty much intact. It's not changed, which means that the mutation is not really having a fundamental effect on the basic properties of synaptic transmission. That said, things get really complicated when you start to look at the effects of networks of neurons and how they communicate with each other, and their really subtle changes can have big effects. And one of the effects that we've seen we've so far just done this in the homozygous mutant animals. We're doing these experiments in the patient model, the heterozygous animals now. But what we see is that in the neurons that are from animals that are homozygous mutants, there is a deficiency in their ability to fire action potentials, to fire spike trains. And this is an important property that neurons have because they need to integrate information they're receiving and decide whether to fire action potentials or not. And the input-output curve, that is how much excitation you give a neuron to drive it to fire an action potential, is changed significantly. You can see on the right graph. It takes more excitation to get them to fire trains of action potentials, and they really never reach the same frequency. So we want to know why that is, and we've heard a lot about GABA A receptors. Dr. Rogowski just talked about their important role, but there's another receptor that we've also heard about, GABA B receptors. GABA B receptors are distinguished from GABA A receptors by the fact that they have much higher affinity for GABA. And when we look at the kind of subtle change in GABA levels that we detect in the mutant animals, we would predict that GABA B receptors would be more strongly influenced than the GABA A receptors, simply because the GABA B receptors have a higher affinity for GABA. And these are kind of complicated experiments. I won't go into them too much for the sake of time, but the takeaway is that we can show that if we activate GABA B receptors with a drug directly, we can get the same kind of effect on the action potential signaling. So essentially, what I wanted to get across with this slide is that we think that the GABA B receptor dysregulation that we detect in the mutant animals might be playing an important role in things like epileptiform discharges. And the reason for this is that things get really complicated when you start to impair neuronal firing patterns, because you have inhibitory neurons and excitatory neurons, and they interact in networks that can lead to things like spike discharges in epilepsy. So I think this GABA B story is going to be interesting to pursue because it might suggest new ways, new paths for therapeutic developments, perhaps. So the conclusions, I will just kind of skip through. I think I've pretty much gone through everything. I wanted to just focus on the bottom thing, which I just mentioned, that these variable changes in GABA B receptors, these high-affinity GABA receptors, may turn out to be an important thing to consider in the disease. And it might be important, too, to think about, as we've heard a lot about variability in patient responses to different drugs and variability in natural history of patients that have mutations, because lots of these SLC6A1 mutations do different things. They're not all acting the same way. And so there may be subtle differences in things like the level of ambient GABA, for example. So we need to keep that kind of effect in mind. So I just want to quickly acknowledge the folks in my lab at University of Montana, especially Greg, who did the electrophysiology, and Allie, who's here, and actually has a poster over here with a lot more of the detailed data on electrophysiology and uptake. And you should feel free to ask her any questions. And I also want to thank SLC6A1 Connect and NIH, the National Institute of Mental Health, and NINDS, who have supported our lab's work on SLC transporters in the past. Thanks a lot.